Welcome to Bio class. Today we will discuss C4 pathway, C4 cycle and photorespiration and some others. Now C4 cycle, you have to know that and you have to compare with the C3 cycle. Okay. So C4 pathway here, C4 pathway here it indicates that C4 oxaloacetic acid as the first carbon dioxide fixation product that they use the C3 pathway or the Kelvin cycle. So, Kelvin cycle that the previous we have completed, that is called C3 cycle. Why it is C3 cycle? Because here the first stable product after taking carbon dioxide, it is carbon number is 3. Okay. Let us take a look that C3 cycle, Kelvin cycle here. Here the carbon dioxide which is taken here, the ribulose 1,5 bisphosphate, and the first stable product here, that 3 phosphoglyceric acid, where its carbon is 1, 2 and 3. So, this is the C3 cycle, C3 cycle. So, now we are going to discuss that the cycle in which that C4 will be there, that carbon will be 4. Let us see that. So, before entering that the cycle, that let us know that here the C4 pathway or C4 cycle, also known as Hatch-Dirac cycle. What is the advantage in this cycle that I am telling you? Here phosphoenol pyruvate carboxylase, it can take carbon dioxide in a very low concentration. It means the carbon dioxide concentration is 10 ppm, then it can take, then also it can take carbon dioxide, but in C3 cycle, Kelvin cycle, ribulose 1,5 bisphosphate, it should have 50 ppm, otherwise it cannot take carbon dioxide. So, it is much more efficient. Okay, this is the first, where this is there, it means that C4 cycle, it is not found in every plant. Where it is found, it is found here, you can see that the corn, the pineapple, sugarcane, Bermuda grass and many other. This cycle, it is found in the mesophyll tissue, outer mesophyll and the bundle seed also. Okay. So, so these things we can see here. Look at here. Look at here. Here it is outer mesophyll cell and the bundle seed cell also. This two different cell is involved, and whereas C3 cycle is only that mesophyll tissue only, it is required. So, these are the I mean difference that we can see afterward. Now, here you have to know that a little bit this part, just look at here. Look at here, Kranz anatomy. Particularly the large cell around the muscular bundle of the C4 pathways, plants are called the bundle seed cells. Just now you have seen, and the leaves which have the anatomy is called the Kranz anatomy. What is Kranz anatomy? We can see this part. Here is Kranz anatomy. Kranz anatomy here indicates that that here the granule chloroplast in the mesophyll tissue and agranule chloroplast larger than normal in bundle seed. So, this type of configuration, it is found in only in C4 plant. Okay. So, C3 does not having this chloroplast, this kind of binding. This is called the Kranz anatomy, typical anatomy for the C4 plant. Okay. Now come here in process. Here carbon dioxide in very low concentration. If it is very low concentration, then it can be accepted here. And here the first stable product here oxaloacetic acid, which is 4 carbon containing, that is why it is called the C4 pathway. So, 4 carbon containing, it is found in outer mesophyll cell. Here this oxaloacetic acid, it turns into malic acid with the help of malic dehydrogenase enzyme. This malic acid enters inside the bundle seed cell. Okay. Enters inside the bundle seed cell, this malic acid in 4 carbon containing, here it releases the carbon dioxide, and here that normal Kelvin cycle, that ribulose 1,5-bisphosphate will accept this. Now here is no problem to accept this carbon dioxide, because now it is not in low concentration, and easily that Kelvin cycle will go on and the food will be produced here in the bundle cell. But here the malic acid, when it gives that carbon dioxide, it turns into phosphoenol pyruvic acid, which is 3 carbon containing, and again it can take that carbon dioxide and it turns into oxaloacetic acid. This is the cycle. It means that the every C4 cycle is having the C3 cycle also, and C4 cycle just the added part of the C3 cycle, and C4 cycle, it shows some efficiency, by taking carbon dioxide, and after that it supplies that ribulose 1,5-bisphosphate as normal Kelvin cycle. Now C3 C4 difference is very important. In your book, one fill in the blank it is given here. You are getting the prepared one, that C3 cycle, that mesophyll tissue, it is there, and actually this Kelvin cycle takes place, and here the cell type, the initial carboxylation is the mesophyll in case of both. There are 2 type of cell here, 1 type of cell. Here the first, this is primary, it is taking ribulose bisphosphate, but here phosphoenol pyruvate. Here the number of carbon in the 5, here it is 3. So first product, that the phosphoglyceric 3-phosphoglyceric acid, and here oxaloacetic acid, and look at here in this way that it is there, and you just can see this part for your information. This all these things it is given. You just go through this, all the difference which is given here. Next is photorespiration. Photorespiration, here we have to understand that the ribulose bisphosphate, it takes carbon dioxide normally, but there are some abnormal cases. Instead of carbon dioxide, the ribulose bisphosphate taking oxygen, because in when oxygen concentration is more, then Rubisco, that enzyme, is greater affinity for oxygen than carbon dioxide. Let us see this this part here. Okay. So ribulose 1,5-bisphosphate, it takes normally carbon dioxide, but in midday when the temperature is high, oxygen content is more, because photosynthesis, when it goes on, the oxygen concentration increases. Temperature high, oxygen content is high, oxygen concentration is high. This affinity for the oxygen is increased than the carbon dioxide. Then what will happen in this condition? The carboxylase acts as oxygenase enzyme, and after that the respiration takes place. So this is the great loss of the plant. Just see that how it happens. So here ribulose phosphate, now it is taking oxygen with the help of oxygenase. Phosphoglycolate it converts into, and it quickly converts into glycolate. Where this is taking place? This is taking place in chloroplast. Now, after that it enters inside the peroxisome. Peroxisome, one unfamiliar that cell organelle that we should know that glycolate. This glycolate again oxidizes into glycine, one of the amino acid. This glycine goes to the mitochondria, and a very less amount of ATP is produced here. This oxidation, the serine, ammonia, all these things is liberated. Ultimately, this ribulose bisphosphate it will be unused for that Kelvin cycle, and instead of that the loss of that I mean here I mean the oxygen is it is used, I mean carbon dioxide trapping is not there, so assimilation is less and food is not produced. Instead of that little amount of energy is produced. So, this is the destructive type of process which is there in the plant. Next one, a factor affecting photosynthesis. Photosynthesis is very complicated process, and the various factors that you know, carbon dioxide, light, temperature, water, all these things, the basic thing it is required. And this requirement according to that the light, it should light intensity, already it is told that the far red and red light it is used, but intensity we can see here in this graph, it should be one optimum, then after that it is the rate of the photosynthesis will not increase if the intensity will increase. So, one certain level is there. About the carbon dioxide, we know that the concentration it should be 50 ppm. Temperature, it is enzymatic reaction, so temperature one should be there. Now, here one important thing that you have to understand that when several factor affects any biochemical process, Blackman's law of limiting factor comes into effect. What is this that I am just explaining to you? This Blackman laws of limiting factor. The Weinert process is conditioned as it rapidity by the number of the separate factor. The rate of the process is limited by the pace of the slowest factor. Suppose here the four different factor which is there: carbon dioxide, water, temperature and the light. So, the pace of that reaction it should be the lowest one. So, suppose here is the lowest is light. On that basis of the light, that temperature, water and carbon dioxide it will be maintained. So it will be controlled according to that. If carbon dioxide is less, then according to that the process it will undergo. That you have to understand. That is the Blackman's law of limiting factors. So, temperature we know that enzymatic reaction, so temperature it should be maintained. The C4 plant responds to higher temperatures and show the higher rate of photosynthesis, while C3 plant have the much lower temperature is optimum. In water, in case of water also, it is in the beginning of the reaction it is required, and water stress cause the stomata to close, hence the reducing the carbon dioxide availability. Okay. So, that is about the factors and all about the photosynthesis. Thank you.